which is our 16th annual tree planting event. The remaining 2 events in our series are the World Water Day, coming up in about 2 weeks, as well as Earth Day in April. So please check our website for details on those upcoming events. Also, if you would like to be more involved in this year's tree planting, please consider sponsoring your tree. We are less than $2,000 away from our $5,000 goal, so everything counts at this point in time. Please refer to a link that Matt's about to drop out into the chat box on how to donate or contact us if you have more questions. This is a great way, I think a really great way to honor someone or have someone memorialized in a tree planting. So please consider that for us. Matt, drop that in there if you would, please. I also want to thank our sponsors for making this possible. First of all, Brother International as presenting sponsor of the Wolf River Restoration Series. And I do believe I saw Vicki Berry sign in, Senior Specialist for Environmental Health and Safety at Brother. I want to say thank you to Vicki and all those folks at Brother. Thank you so very much. Also, International Paper is the event sponsor for our 16th annual tree planting. And James McDonald is with us tonight, the Director of Global Citizenship from International Paper. And we want to say thank you to James and all the folks at IP there. Additional sponsors are Bank of America, Buckman, Chris Hill Construction, Crawford Howard Family Foundation, Griffles Foundation, and FedEx. Thanks for all of you for making this possible tonight. Now let's hear a quick message from Don Cummings, the president of Brother International. Nick, if you'll help me out here.
Hello, I'm Don Cummins, president of Brother International. At Brother, our mission is to live our at your side promise and to simplify and enrich the lives of our customers, employees, and communities. For many years, we have stood with you at local in-person volunteer events to protect and steward important landscapes and healthy habitats. It's with great pride that we continue to support the Wolf River Conservancy in new ways by presenting the 2021 Wolf River Restoration Series. This series of events is designed to provide fun, interactive, and educational programs dedicated to restoring and conserving our ecosystem. We hope you enjoy today's program.
I know we can't call it everyone, but I did just see Beth Bazar join us from Buckman, who are also one of our sponsors. So I see y'all, I'll try to catch y'all out there. But right now, I want to turn tonight's program over to James McDonald, who I mentioned just earlier from International Paper, to give us a little introduction from IP and also introduce our really good speakers for this evening. It's going to be a great lecture, you guys. Thanks so much for joining us. James?
Thanks, Mark. International Paper is really excited to be one of the sponsors for this event. As most of you know, International Paper is basically a natural resource company, and we really depend on healthy and abundant forests around the world and in our backyard. Programs like this, and partnerships like we have with the Wolf River Conservancy, help International Paper achieve its vision of becoming one of the most successful, sustainable, and responsible companies in the world. Now it's my great pleasure, excuse me, to introduce Drs. Craddock and Laporte. Dr. Hill Craddock is the UC Foundation Davenport Professor in Biology in the Department of Biology, Geology, and Environmental Science at the University of Tennessee in Chattanooga. Having grown his first chestnut tree from a seed planted at age 15, Dr. Craddock's current research is focused on the restoration of the American chestnut to the Appalachian hardwood forest ecosystem and the establishment of a commercial chestnut industry in Tennessee. His project areas include breeding for resistance to chestnut blight and root rot, chestnut germplasm collection, and chestnut cultivar evaluations. Dr. Robert Laporte is an assistant professor in biology at Rhodes College. He grew up on the south coast of Oregon, gaining a passion for studying species interactions and evolutionary dynamics while rambling through the forest and coastal ecosystems. While Dr. Laporte's current research focuses on the genetics of the creosote bush and its relatives in South America, His recent move to Rhodes College in 2018 reinvigorated his interest in the American chestnut tree. With the help of some local naturalists, he began searching remnant forests in southwestern Tennessee and northern Mississippi to better understand the distribution and genetic diversity of American chestnuts to aid breeding and restoration efforts. I'll now turn the program over to Drs. Craddock and Laporte. Thank you.
Hi, let me begin by sharing my screen. And starting my PowerPoint presentation. I can't see you out there, but Kathy told me there are more than 100, maybe 130 participants at this event tonight. So I'm very, very honored to be here, and I want to thank the Wolf River Conservancy for honoring me with an invitation to talk to you tonight. And this is also a great opportunity for me to work a little more closely with Rob Laporte, who I've known a little bit at a distance over the last couple of years. We've been aware of each other's work, but we've only just recently begun to work together on this project. So I'm going to share with you tonight some of my work on the American chestnut. I'm going to take a little bit of a step back and tell the story of the chestnut tree, because I want us all to be on the same page when we begin. Chestnut trees are part of a large, important group of trees in the Fagaceae. They're related to oaks and beeches, and so they form a part of a vast community of trees that covers the northern temperate zone. The genus is Castanea, and depending on the author, there may be something like 8 species or maybe up to 13 species. Some authors have considered even more. There have been trends in botany over the last 100 years of lumping similar species together into one diverse species. Other authors have split the genus into multiple different species. So depending on who you believe and who you read, and by the way, don't believe everything you read or hear about chestnut trees. We have a group of species in East Asia. One very distinct chestnut tree from Japan, Japanese chestnut, Castanea crenata, that will figure later in my talk. So that species from East Asia, from Japan, actually not very closely related to the Chinese species, of which we have maybe 3 or 4 types. The one we call Chinese chestnut is Castanea mollissima, occurs across a broad range in China. And then 2 smaller, in terms of their range, two less well-known species, Castanea henryi and Seguinii, Castanea seguinii. Seguinii is a shrub, and henryi is a tall forest tree that's important also for timber. And it's used for its edible nuts as well. In North America, we have 2 or 3 species, depending on the treatment. You can see that the author of this map labeled on this part of the map, C. dentata, the American chestnut. And then the other species is Castanea pumila, which is the chinkapins. Now, some authors have divided the chinkapins into 2 groups, 2 kinds, 2 botanical varieties, variety Ozarkensis or variety pumila, C. pumila, variety pumila. Some authors have elevated the Ozark chinkapin to its own species, which would be Castanea ozarkensis. And it's interesting because it's a area of natural occurrence, is a tiny part of North America, which is limited to the Ozark uplands in northern Arkansas and southwestern Missouri. The most famous of our chestnuts in the United States, of course, is the one we call the American chestnut. And it historically was a big, long-lived, important timber tree important for many, many uses for the lumber, but also for non-timber uses, particularly as a food source for humans and for all other animals that lived in the forest, passenger pigeons, deer, bear, other birds, arthropods, and insects and other arthropods depended on it. Much of what's written and much of what I read today about the American chestnut suffers a little bit from, let's call it, hyperbole. It was a fantastic story, a fantastic tree, and it seemed to get a little better, a little bigger, a little more numerous every retelling. So think about it a little bit. There have been some excellent papers recently trying to document the pre-blight abundance and distribution of American chestnut. It certainly occurred across a very large geographic area, but within this large geographic area, it was not uniformly distributed. So for instance, in the south and in the southwestern part of its range, on this map, that's the blue area on this map. It occurred, but it wasn't the dominant species of tree. I mean, there were far more pine trees down here than there were chestnut trees. And there may have been some areas in the southern Virginia, Southwest Virginia, Western North Carolina, where chestnuts truly formed a solid, dense canopy of solid chestnut. But again, much of what I read on the internet about American chestnut is an exaggeration in terms of the redwoods of the East, or 1 out of every 4 trees in North America was a chestnut. No, they're not redwood trees. They're nice big trees, like an oak tree is a nice big tree, but they're not redwood trees. And 1 out of 4 trees in the United States was not an American chestnut. They did grow quickly, and on some sites, they were the fastest tree. They could keep pace with tulip poplar. They could grow as fast as white pine on some soils. They were fantastically useful because the wood could be split. It didn't require a sawmill, so it could be used on the frontier. You could, with a wedge and a maul, you could split it out. The tree on the left in this photograph was actually part of a timber yield study. It was 110 feet tall. It was 80 years old, and it was grown on abandoned farmland in Connecticut. The photo was taken about 1914, which would have been right about soon after the appearance of chestnut blight. The photo on the right, even though it looks older, is about from the same time, and that's a picture from Western North Carolina. This is a big old tree, may have been 300 years old. It could have been 500 years old, and there's a person in that figure. I'm just going to tap on the screen so you can see the human for scale in that photograph. That was probably typical of the Western Carolinas, Southwestern Virginia, East Tennessee, large, old, very, very long-lived trees. All of that came to an end in the early 20th century when a new disease was discovered. On trees in New York City, there were some American chestnut trees, very old at the time, in the Bronx, at the zoo, the Bronx, that died. And a plant pathologist from Brooklyn went all the way over to the Bronx to look at these dead trees and saw a fungus living on the bark. It was a fungus apparently unknown to science. He described it and named it Diaporthe parasitica. Later, the name was changed to Endothea parasitica, now Cryphonectria parasitica. Don't get hung up about the names, but the fungus Cryphonectria parasitica is a terrible pathogen. It kills the American chestnut trees very, very quickly. Soon after its discovery, it was hypothesized that this fungus came from Asia on imported nursery stock, we now know, from Japan. Chestnuts were popular in the East Coast of the United States as a nut crop. The American chestnut makes a tiny little nut. So the Japanese chestnuts were popular as an orchard tree because they make a really big nut. So on this nursery stock that came from Japan was this fungus that causes a disease on the Japanese chestnut, a bark disease, but doesn't kill the tree. The American chestnut had no resistance, so highly susceptible. It eats the cambium, which is the living tissue inside the inner bark. It girdles the tree and quickly kills it from that point up. So a large full-grown, mature American chestnut would be dead within one growing season from infection. Some smaller trees could linger longer, but effectively caused a pandemic in the true sense of the pandemic, that there were no unaffected individuals. Within 50 years, this disease swept through the range of the American chestnut and completely removed the American chestnut tree from its ecological niche as a canopy dominant tree. There's probably no worse forest disease in history, a pathogen that so quickly and completely wiped out its host. And in fact, in response to the chestnut blight, the government, the United States government, enacted its first laws on plant importations, restrictions on plant importations to control the importation of diseases and exotic invasive pathogens that would damage agriculture and forestry in the United States in response to the chestnut blight pandemic. It's difficult for us to imagine the scale and the scope of this disaster. Billions and billions of trees over thousands and thousands of square miles of land completely destroyed. The trees are not extinct though. The fungus kills the bark and the above ground part, but if the tree is lucky, the fungus won't grow underground, not because the roots are special particularly, but because the soil community is antagonistic to the fungus. And so we still see, and I'm going to jiggle the mouse here. This is Jonathan Pruitt, a student of mine who has found an American chestnut on the Cumberland Plateau. They persist as sprouts from the base. In other words, the root crown, which is the area of tissue, meristematic tissue at the junction of the root and the shoot, that root crown continues to shoot, produce new shoots, even 100 years now after chestnut blight. So we find them occasionally. This specimen here, I didn't do it on this one, but a colleague of mine, Joe Shibig from Middle Tennessee, sampled trees in the Cumberland uplands that had about a centimeter of diameter, about a ½ inch diameter, had 35 growth rings. So a tree this size, which most certainly was alive, by the way, when blight hit in the 1930s, blight came through Tennessee in the 1930s, has grown so slowly that in 35 years, it took 35 years to make a stem a ½ inch in diameter. The trees on the right actually all arise from a single stump. I love this picture because Walter and Christine are standing there showing you, it's sort of, you can just imagine that semicircle of sprouts coming up from a blight-killed stem. Now those trees have blight. In fact, Christine is pointing to a canker. It's a superficial canker caused by a hypovirulent strain of the fungus. This is a story, you'll have to invite me back to tell the story of hypovirulence, but it's a potential biocontrol for chestnut blight that we've used effectively in our breeding program. It works naturally in a few places in North America, in Michigan, for instance, and it's widespread in Europe now. The European chestnut also susceptible to chestnut blight. But by the 1950s and early 1960s, a mild form, the hypovirulent form of the blight, began to attenuate blight symptoms at the landscape level. And so this hypovirulent strain of the fungus is actually caused by a virus. So there's a virus that attacks the fungus and keeps the fungus from killing the tree. Just like the enemy of my enemy is my friend, right? And so the virus, this hypovirus, attenuates the pathogenicity of the fungus, prevents it from growing. I'm going to just check something real quick here. Kathy, if you can hear me, tell me you can hear me and that everything's going okay.
All good.
Matt, thank you. I can hear some chatter in the background, which makes me think that somebody's not muted. I'm just, that's the professor in me talking now. Which brings me to what I do for a living. Of course, I'm a teacher, but I'm also a researcher, and my research is focused mostly on the American chestnut. as James said in my introduction, I planted my first chestnut tree at age 15. I've been interested in chestnuts my whole adult life. And I feel very lucky to be able to work here in Tennessee, which is close to the world center of chestnut diversity. Breeding for resistance to Cryphonectria parasitica. Now, of course, Cryphonectria is the fungus that causes the chestnut blight disease. We can take advantage of this ability of the chestnut trees to resprout from the base to breed them for blight resistance. Now, the plan, not my plan, the idea was hatched years ago, some 30 or 40 years ago, by plant breeders following a classical plant breeding strategy that we used in corn and tomatoes to bring disease resistance into the varieties that we grow by making hybrids with other species. And so if you have a tomato you like or a corn that you like, but it has a disease, you might find a wild relative of that crop plant that has resistance to that disease and make a hybrid. And then by a series of what we call back crosses, we can recover the tomato we like and maintain the disease resistance. And so this process is called back cross breeding. And in evolutionary terms, or in ecological terms, we call it introgression. And so the genes from one species then are moving or introgressing into the genome of the other species. Every generation dilutes the genome of the foreign species by 1/2 and doubles the amount of the genome in the recurrent species. And so if we start with the Chinese and American hybrid at the top, the F1 hybrid is 50-50 American. The first back cross is 3/4 American, 1/4 Chinese, and so on and so forth. It really doesn't work like that because real life is a little more complicated. And the idea is that by the time we get down to a tree that's about 15/16 American, it would be indistinguishable from the American tree in all of its adaptive characteristics and what it would look like, how it would grow in the forest. But if we do this correctly, we can select for trees at every generation that have the blight resistance. Eventually, we end up with true breeding blight-resistant American chestnut trees. I'm going to tell you right off the bat that it's been more difficult than we thought 25, 30 years ago because the genes for blight resistance are actually many, and they are scattered around the genome on multiple different chromosomes. So it turned out it was more complicated to breed for disease resistance in chestnut trees than it was for certain diseases in tomato or corn, where we had a single locus on a single chromosome. It was relatively easy. So just very quickly, some slides to share with you the kind of work it is. Of course, here's an American chestnut. This is a naturally occurring wild American chestnut that has blight. It's had blight for many years, but it hasn't died because it's infected with that hypovirulent strain, and it's in an ideal location. It's on a ridge top next to a pasture. Lots of other issues going on with this, but the tree was still alive. It was that's me in the bucket there, so I didn't take this picture. The flowers, from a botanical point of view, it's a monoecious species. It has both male flowers and female flowers on the same plant, but they are not self-fertile, so they require cross-pollination, cross-fertilization. The female flower is in this, I'm jiggling the mouse here, I don't know if you can see, in this little involucre, which becomes the burr, the prickly burr of the chestnut. And the true chestnuts, like Castanea dentata, the American chestnut, have 3 nuts per burr, parentheses. The chinkapins have just 1 nut per burr. And that's important if you're trying to identify an American chestnut in this part of the world, because you might find a chinkapin and they look superficially very, very similar to the American chestnut, except that on certain botanical characteristics, for instance, the number of pistils per cupule, the number of nuts per burr differs. 1 nut per burr in the chinkapins, 3 nuts per burr in the American chestnut. The male flowers are held in this fuzzy inflorescence here called a catkin. And that's hundreds and hundreds of tiny male flowers that produce the stamens, the anthers, where we get the pollen. The pollination is very straightforward. We can collect the pollen, we can dry it, we can freeze it, we can store it from year to year. We put it in little vials. We transfer the pollen with the lid of the vial to the receptive surface of the female here. We put pollination bags over the female flowers to prevent unwanted pollination. So we think we know who the dad is. We also do controls. This photograph was taken at our research farms. I work with the American Chestnut Foundation. I'm wearing my Chestnut Foundation vest. I don't know if you can see. I work with the American Chestnut Foundation is a nonprofit that is dedicated to restoration. We have a research farm in southwestern Virginia. The seeds then are harvested in October. The American chestnuts ripen in October. We stratify them in cold and slightly moist peat moss for the winter, plant them in our greenhouses, or we can direct seed. If we have a nice seed bed prepared in the orchard, we can plant the seed directly into the orchard, but mostly we plant them in the greenhouse. We transfer them then to the nursery where they get attention, they get water, they get fertilizer, they get sunlight, they grow. We transplant them into orchards. And I should mention that this is almost entirely citizen science. It's done by volunteers. Almost all of the labor is volunteer effort. We have volunteer landowners, we have volunteer growers, we have volunteer planters, pickers. It's a marvelous example of citizen science. We have people out in the woods looking for chestnut trees at every level. We give them tender, loving care. We grow them, we water them, we protect them from deer and cattle, from weeds. And when the trees get about maybe 2 or 3 inches in diameter, we kill them as fast as we can. And I use the term screening. Screening is a euphemism for killing the trees. To make any progress in the breeding program, we have to separate the susceptible trees from the trees that are potentially carrying the genes for blight resistance from their Asian ancestor. Maybe I forgot to tell you, but I'm using the Chinese chestnut and the Japanese chestnut as the species, the resistant species. The Chinese times American or a Japanese times American hybrid, running that through the backcross program. At every generation in that chart, we have to select. Only the very best trees will be advanced to the next generation. So the inoculation is fairly straightforward. We grow the fungus on a potato jelly in a petri dish. We take a plug of bark out of the bark. We replace that plug of bark with some mycelium from the fungus, and that takes a lot of effort that we do in the hottest part of the summer to make it more interesting for the volunteers. Here's my volunteer crew. It was really hot that day. If you're from Memphis, you know about hot. If you're watching this from New York, you don't know about hot. The Everclear, by the way, is to sterilize the tools. I just want to point that out. We can also do the screening on yearling trees, on seedlings grown in the nursery. We've been trying this method now for several years. It would save a lot of land, it would save a lot of time, it would save a lot of space, it would save a lot of money, but it doesn't work. And I mean, I can kill the trees. The problem is it's a very blunt, I don't have any resolution in this asset. So the small stem is so susceptible that I can kill all of them, even the resistant trees. In fact, the photograph on the right here, this is the Chinese chestnut control that is the resistant tree. This fungus, this pathogen is so devastating that it, on a tree this size, it will kill it regardless. In other words, to have the expression of resistance, I really need a tree that's at least 3 or 4 inches in diameter or bigger, 5 or 6 inches in diameter before I can detect signs of actual resistance. Anyway, to measure resistance, we measure the cankers as they grow. This is a susceptible tree, it's dead. The resistant trees form a wound tissue, a callus. This one was inoculated with 3 different strains of the fungus that vary in their virulence. The top 2 are hypovirulent strains. The bottom one is a fully virulent strain. That same tree photographed about 2 years later has completely healed over. Even the canker caused by the virus-free virulent strain. This is the lethal strain that killed its sibling. These are full sibs. This one didn't get it. This one got it, has now healed. So this is a sign of high level of resistance. The trees that don't make the cut get removed from the orchard. And because these are large trees, they're heavy, it's a huge effort. the value of the one we select, weirdly, is proportional to the number of its siblings that we selected from. You know, the best tree out of a group of 10 or 20 would be a pretty good tree. The best one out of hundreds or thousands is probably a very good tree. When we then advance these trees to the next generation, when we get all the way to that 16th tree or 32nd part American, it's essentially an American chestnut with blight resistance. We can do intercrosses, and the intercrosses are designed to fix the resistance so that all of the offspring would be resistant. We're not there yet. So here's a photograph showing us preparing the intercrosses. These are hand pollinations. You can see the pollinations. I've got ladders, I've got a bucket lift, I've got volunteers. Eventually these trees get planted, these later generation trees get planted very densely because we have a very, very high level of selection. In other words, I really only want one out of every 150, or I'm really looking for the best one out of 100 at this point. So I can plant them close together because I know we're going to select very ruthlessly to get the good ones. And this is to fix disease resistance so that all of the offspring will be resistant. Of course, this takes a lot of effort. Here's the planting crew. You may have heard, so this describes the classical plant breeding methods that I use. You may have heard about the transgenic approach. There's a group in New York that we've been working with very closely for the last 25 years at the State University of New York at Syracuse at the Environmental Science and Forestry Lab up there, led by Bill Powell. And Bill Powell sent me these slides. They're using a gene from the wheat plant. And this gene is an oxalate oxidase. It destroys oxalic acid. And oxalic acid is one of the chemical weapons that the fungus uses to attack the plant. So if you destroy oxalic acid in the plant tissue, you're essentially disarming the fungus. Okay, and it's been very effective in greenhouse trials and limited field trials. It's highly regulated though by EPA, USDA, and FDA, because chestnuts are a crop, chestnuts are a food crop, and this is considered a pesticide by EPA. It needs to be registered, and we're going through that process now. That's another lecture. But I did want to mention to you that this strategy is complementary to the breeding strategy. And even if this works, and even if it gets deregulated, the tree that was genetically transformed is a single individual from New York State, which means not suitable for planting in Tennessee. Not because we don't like, I know I've said two things about New Yorkers now. One, they don't know about heat, and two, we don't want their chestnut trees. Not because I don't think they're good trees, it's just because I don't predict that a New York American chestnut will have the adaptive characteristics that it needs to do well in the South. And we know this from lots of other examples of other tree species. When we move trees far from their latitude of native occurrence, they are maladaptive, they don't grow well. So better for us to look for local germplasm. In other words, we need to find and conserve the genetic diversity of the southernmost and the southwesternmost American chestnut trees, because those are, there is where we think the genes that we need to grow well in the South, particularly in the face of the predicted climate models that, 50, 100, 500 years from now, following the current trends of global warming, how American chestnut range will move north. These adaptive traits in the southern populations may be even more important in the future. Eventually we get to forest type plantings, and we are doing this now in collaboration with private landowners and with the Forest Service. These are what we call progeny tests. The progeny test can be established by transplanting or by direct seeding, and we've done that. I don't have time to report on that. I also don't really have time to tell you about the next disease. We're breeding for two diseases. The other one is the Phytophthora root rot, caused by
Dr. Craddock, excuse me, just wanted to give you your 5-minute warning.
Thank you, Kathy. I had this idea, I was running out of time. I'm just going to tell you then that we're working as hard as we can on this other disease as well. This one, terribly, probably more destructive than the chestnut blight, in the sense that it kills first the roots. So when the trees die of the root rot disease, they're gone. There's no chance for resprouting. And the microorganism, this terrible pathogen, persists in the soil, which means the presence of it in the soil precludes planting American chestnuts forever in those soils. So we're using here again classical plant breeding methods. We're very careful. We've collected isolates from local trees. I'm going to go back up 2 slides. This tree, this is a whole row of dead hybrids died of Phytophthora. There's one living tree in the background. That's a Japanese American F1. That tree became very important later. There's that same tree several years later. We were able to use it as the pollen parent and as the seed parent in crosses that we made. We plant these things in death chambers. These are the killing chambers. We inoculate them. I use this as curriculum for my students. Here's my mycology students. We make selections. Here are the phenotypes. The top left is a healthy tree. These two are intermediate levels of resistance. They have necrotic lesions on the roots, but they're going to die probably soon. And of course, then this phenotype is completely dead. These trees were planted out into orchards where we knew the trees were dying of Phytophthora. And so the selected trees, the ones that we had screened in our tubs there, in our killing chambers, have grown well and are showing good vigor and persistent resistance. Now this tree is only 2 years old. Since it's been in the field, it's been 2 years in a pot. But this is an orchard in Middle Tennessee that had suffered from the Phytophthora root rot. And so my last slide is sort of a summary of we're breeding for blight resistance, we're breeding for root rot resistance, but all of this depends on having access to the plant genetic resources, to what plant breeders call germplasm. So the germplasm is out there, we think, but to use it, we have to find it, we have to know where it is, and we have to know something about it. What species is it? Is it American chestnut? Is it a chinkapin? Is it already a hybrid? Is it a Chinese chestnut that's escaped from someone's backyard? We need to characterize that. We can use molecular biology tools now. We've got DNA fingerprints that we can ask questions about the diversity. And then we can conserve that diversity. And my idea is that we can conserve the genetic resources by capturing those genes in the form of blight-resistant hybrids using classical plant breeding or using the genetic engineering approach, so that we can conserve this vast amount of genetic diversity that we know exists in the South. More so for lots of other reasons we didn't have a time to talk about. The genetic diversity of our southern populations is much greater than that of the northern populations. We can also propagate these trees by grafting. Again, that's another lecture. We grow those grafted trees in orchards together, and then we can breed them and advance them. And ultimately, we want to conserve these trees either in the forest by preserving large areas of natural habitat of key importance, or in orchards in backyards, which are, again, complementary strategies. That's my last slide, and I'm going to turn it over to Rob now. Rob, I went about 8 minutes over, but you're up.
Right. Thank you, Hill. That was great. And let's see, I think maybe you just need to hand over the sharing to me.
Can you, Hill, could you stop sharing your screen by going up to the top to stop sharing?
Looks like you did. So I'm going to go ahead and put mine up here. Is that showing up okay? All right. Well, thank you, Dr. Craddock, for a really great talk. And I want to say it's really a pleasure to be here tonight, to have this invitation, this opportunity to speak with everyone. So thank you for taking the time tonight. Thank you for everyone at the Wolf River Conservancy for organizing this talk, and all of the sponsors for making this talk possible. So my name is Rob Laporte. I am a faculty at Rhodes College. I'm an assistant professor of biology. And in my lab, we're really interested in studying biodiversity. We're really interested in like what constitutes biodiversity, the origins of biodiversity, and also the interactions among different units of biodiversity. And one of the things that has really fascinated me about this American chestnut story is that it really represents the loss of biodiversity, or the extinction of a species. I mean, American chestnut has become functionally extinct over time because of the chestnut blight that Dr. Craddock talked about. But I want to go back to that story and remind everyone about how important American chestnut was for a long period of time. I mean, this was a pretty common species. It was a dominant canopy tree throughout Eastern North America, and especially in the Appalachian region, all the way up into the Northeastern United States. But you know, it was important for timber practices. I mean, this was a large tree, especially in the Southern Appalachians, where it could become quite large, like some of the pictures that Dr. Craddock showed. It was important for lumber, but it was also really important as a food resource for people, but also for wildlife because of these yearly productions of large, fatty, nutritious seeds that were dropping to the forest floor and eaten by vertebrate organisms. But it also was important for invertebrate species. So a lot of pollinators visited this tree. It produced these massive blooms, these floral displays of male flowers that were out there. They could go collect pollen from those and nectar. But then it was also really important for nutrient cycling in the forests, right? So these eastern hardwood forests were dominated in some places by American chestnut. It was dropping leaves every year. Some of these large trees were locking up carbon for tens or for decades or centuries at a time, and then they would fall and slowly decompose. And you know, that was a really important component of the forest until the introduction of chestnut blight. It was also, currently it's also being lost, or it's absent from those forests. And we know that these eastern forests in North America are starting to recover from land use changes that have occurred due to increased urbanization, especially starting in the mid-1900s, but also land use changes with cessation of land clearing that was associated with small farms and small production. So now we see here on the left this map showing this green coloration, which is really showing the intensity or the amount of woody biomass that occurs in these forested regions of the eastern United States. And you'll see these darker colors, these darker green shadings are associated with the historical distribution of American chestnut. But what's problematic about that from my perspective and for larger perspectives as well is that these forests are coming back in these mountainous regions and throughout the eastern United States, east of the Mississippi River especially, but they're lacking a really important historical component because of this blight decimating these trees as it moved out of the New York City area in the early 1900s and progressed ruthlessly southwestward through the range in these waves, you know, every decade. And by the mid 1900s, by 1950 or so, it had reached that southwestern extreme of the range. And you couldn't find a place in the natural range that was unaffected by this ruthless fungus that kills the trees that it encounters. And so what this sets up is this real big change in the ecosystem dynamics of these eastern forests, right? We go from having these, you know, in some places, enormous trees, these really fantastic canopy trees, dominant trees that are really controlling nutrient cycling, providing habitat for species that are being used by humans that are occupying these places. We lack these big trees. We lack these inputs of nuts. We lack these inputs of woody debris and leaf matter. We now are stuck with small understory, shrubby looking trees that really have a different ecological role. So we're left with a functionally extinct species that really isn't becoming reproductive on the scale that it once was historically. And so today it's considered in many places to be a rare species or even a threatened or endangered species. Here in Tennessee, the Tennessee Natural Heritage Program includes American chestnut, Castanea dentata, on the rare plant list. In the state of Tennessee, we consider this a sensitive or a species of special concern or a sensitive species, a S2, S3 rank, if you're familiar with these, basically meaning that it's relatively rare throughout the range and it's persistently threatened with extirpation or extinction due to this recurrent blight infection that occurs. I mean, the blight is resident, it is here, and it will infect these trees. And so they could become extinct at any time. And so at a global level, global non-government or governmental organizations consider this to be relatively stable or apparently secure or stable. However, in some parts of its range, at least, it is threatened with extirpation. What's interesting about the Tennessee Natural Heritage Program's designation here is that they're really only considering the range of the species as occurring in Middle Tennessee and eastern parts of the state as well. So here in the Western Highland Rim, the Central Basin, the Cumberland Plateau, and the Blue Ridge Mountains part of the state. But they aren't really considering this far western component of the state, or the far western part of the state that includes the coastal plain where we are down here, or even the Tennessee uplands a little bit further east of us. And that's kind of problematic from my perspective as well. And Dr. Craddock alluded to this because there's a lot of biodiversity down here, which I'll get to in just a moment. But we know, for example, that chestnut has been in this part of the state, or was once an important component of the forests of this part of the state. So one of my colleagues, my co-investigators, who's been integral in studying American chestnut in this part of the state, in the southwestern part of the state, has also done a bit of literature research and dug up some examples, some really cool examples of mentions of chestnut occurring in Western Tennessee, going all the way back to the early 1800s, where people were definitely interacting with the tree. They were out to collect the nuts from these trees to eat, right, as supplemental component of their diet. And so some of you may be familiar with this Old Times in West Tennessee, sort of a semi-historical reminiscence of these Western pioneer days, non-Indigenous pioneers that settled parts of West Tennessee. And there's mentions in here of going out chestnut hunting. But more than that, they even give us some indications of the ecology of the trees. They occur on these bluffs, these upland bluffs above the rivers where they were searching for these chestnuts, for example. And this occurs in somewhere along the Hatchie River near Durhamville, Lauderdale County, or perhaps at the border with Haywood County. And then my co-investigator also found this diary entry. So this was a diary that is cataloged at the Fayette County Library in Somerville, Tennessee. So in 1864, October of 1864, Hattie Fraser, a young woman at the time, right at the tail end here of the Civil War period, mentions having some visitors over, and then they went out and they hunted for some chestnuts, presumably to eat, as sort of a pastime thing. He recently sent this to the American Chestnut Foundation. This is coming out now, or is printed now in the winter edition of the Chestnut magazine produced by the American Chestnut Foundation, if you are familiar with that or subscribe to it. But I want to come back to this idea that here in the southwestern part of the species range, the southwestern part of Tennessee, in fact, or even northern Mississippi, American chestnut is super important from these conservation and restoration efforts. It's also important from just a biological, ecological, or evolutionary perspective as well, because the southern part of the range harbors a massive amount of genetic diversity compared to these northern components, or these northern portions of the range. And this genetic diversity is really crucial for these breeding efforts, or even the transgenic efforts, because we need to breed trees. We need to reestablish trees that have this genetic variation. As Dr. Craddock said, if you take a tree from Syracuse, New York up here, and try to introduce it to Tennessee, it's probably not going to deal real well with summer lasting from April until November, right? And conversely, taking a tree from Tennessee, it may not do real well if winter lasts from November until April, right? Or even May in some cases. And so this local adaptation that can arise from this genetic diversity is super critical for these efforts. It can also help us deal with the trees as climate is changing. If summers are starting to warm throughout more northern parts of the range, having trees that are adapted to that, having genetic variation to deal with that variation of those more intense summers is going to be really critical. Now you might be asking, why is there so much genetic diversity shown by these orange dots here in the southern part of the range versus the northern part of the range? And this really goes back to one of the major hypotheses, or one of the major drivers of this being that the North American continent was covered with ice, a mile of ice for a long period of time. So we can go back to the last glacial maximum period, what we call the Wisconsin glacial, about 18,000 years ago was the peak of that. And you can see that most of North America was covered, and that glacial lobe penetrated all the way down here into the Great Lakes region, the northeastern US, as far south as even parts of southern Illinois and southern Indiana. So not too far north of us here in West Tennessee. You might be saying, well, that's great. Trees could live here where the ice is not, I suppose. But what we know is that ice-free region was tundra. So this is habitat that you would be encountering if you were in northern Canada or northern Alaska, for example. This is a vast treeless plain with low-growing plants, right? And there's not trees there. It's defined by the lack of trees. So this was a pretty harsh, cold environment at the time. And chestnut was nowhere to be found. And we know that those areas were tundra along the ice zone or the ice front from pollen records. So these are records of sediments, or these are sediment cores extracted from lakes that have been around for a long time. And looking at those sediment layers, you can recover the actual pollen that's in there. It's been preserved in these sort of anoxic environments in the sediment, and identify what species of pollen that is, or what species of tree that pollen came from. And one of these pollen records that's pretty well studied here in the South comes from White Pond, South Carolina. And it's a few hundred miles from the Mid-South or from Memphis, but today occurs in a very similar climatic zone as the Memphis area, the Mid-South here. And so probably at least gives us a first approximation or a rough estimate of what the climate was like here and what trees were growing in the Mid-South region. And what those records tell us is that going back about 10,000 years, as that glacier was starting to retreat, that we had species like Picea, so these are spruces, and we had a lot of pines around that time as well. These are species that are more typically associated with boreal forest or taiga. And so that's what you would encounter if you went up into sort of the middle part of Canada or Alaska. And we don't start seeing our typical hardwood species or deciduous tree species that we associate with forests around the Mid-South now, or even throughout most of Eastern North America. Until much later. So certainly not until about after 8,500 years before present, and really not until they don't become really common in the pollen record until about 6,000 or 7,000 years ago. That's when we start seeing things like quercus. Those are the oaks. We see elm pollen in the record as well. We also see fagus. Those are the beeches. So these are the oaks and the beeches are actually relatively close relatives of American chestnut. We see maples as well in those records. And if you're wondering what those records are, how they get created, right? So, you think about if you've been out on a spring day or something and all that pollen from the pine trees is being cast off, most of that pollen misses its mark and lands on the water, gets washed to the shore or sinks to the bottom of that lake. And, that pine pollen is pretty characteristic. It can be identified under a microscope. And so once that gets buried by some sediments, we can say, oh yeah, there's a lot of pine pollen 8,000 years ago, but less of it 6,000 years ago. And then we start seeing some more of it more recently. And so that's how those are interpreted. But those pollen records are really important because what they tell us is that, yeah, there was tundra to the north of us here, right along that ice area, the ice face. But then here in the Mid-South and throughout a large swath of Eastern North America, it was actually taiga-like forest. So as I said, this boreal forest where we have some of these conifer species. There were some hardwood species or deciduous species in there. And you might be thinking, yeah, Memphis looked like this bottom picture just a couple of weeks ago. So what's the big deal? Well, remember that this is the type of habitat that you would see up in Canada. So imagine if it looked like this, if it was that week-long period that we experienced here in Memphis, but it lasted for 4 months of the year and it was every year, right? This is a harsh winter and it precludes a lot of these species from inhabiting that type of environment. So where was chestnut? The leading hypotheses suggest that it was pushed even further south into these sort of temperate conifer mixed forests that were in the far southern parts of Mississippi and Alabama along the Gulf Coast, or even potentially parts of eastern North and South Carolina on the coastal plains, and probably really even out onto the continental shelves, which were exposed at that time because of the changes in ocean levels or sea levels. The sea level had dropped during these glacial periods, and so more land was actually exposed. And then as that glacier started to retreat, the species in those forests, those really southern areas, started to expand northward. And as they expanded northward, those trees, for example, of chestnut that expanded northward carried with them less genetic diversity than the trees that kind of stayed behind. They expanded so quickly, they subsampled the population. They took fewer of the genetic variants there and expanded northward. And so we end up with this situation where these orange dots are showing populations that have more genetic diversity, these blue ones, less genetic diversity. But one of the things you'll notice here is we've kind of got this hole in the map, right? So these far southwestern parts of the range here in southwestern Tennessee and northern Mississippi are not really represented in these studies of genetic diversity. These past studies, they sampled these latitudinal gradients, but they left off, these far western portions of the range. And part of that is just practical. Chestnut was here, as Dr. Craddock said, but it probably wasn't the dominant species. It wasn't the most common. And that's shown in this map here. These lighter green areas are what's considered the extended range, or parts of the range where it occurred, but it wasn't super common, like these darker portions of the range where we have chestnuts being sometimes dominant species or even growing in these big groves. And so we know that, that lack of inclusion is really sort of a methodological choice. We know that the chestnuts are there from those historical records, but it also is probably a sort of just a occurrence due to just luck, right? Here in the Mid-South, we're kind of removed from some of the major population centers that we see on the northeastern coast, right? We're not part of the megalopolis corridor from Washington, D.C. up through Philadelphia and New York City and all the way up to Boston, where there's a lot of people that get out of the cities and go and hike in the woods, and they know about chestnut and they go out and find it. We're also pretty far away from the Appalachian Mountains, where a lot of people go and recreate. So people go out to the national parks there, they sort of bypass Memphis or just drive through on their way to the Great Smoky Mountains, and they're likely to find chestnut there, but they aren't going to find it very often here in the Mid-South. And so with some co-investigators of mine, principally David Smith, who's a local naturalist and has been really involved with the American Chestnut Foundation, the Conservancy, has been doing a lot of work here in the Mid-South over decades to really identify not just chestnut, but many other species of concern or interest here. And then Julianne Ng, who's been working with me, we've been trying to fill some of these gaps by finding chestnuts in this southwestern portion of the range. And we recently published a paper in the journal Castania to document some of our findings here, where we've been finding these populations of trees, small populations here in Fayette County and Chester County and McNairy County. We've recently also added a couple more locations where we found some other trees on part of the Wolf River Wildlife Management Area in southeastern Fayette County, but also near the headwaters of the Wolf River, actually in Benton County, Mississippi, in the Holly Springs National Forest in the Baker's Pond unit there. And so we're really working to try to find these things, but what do they look like? When we go out there and we find these trees, what we're really finding are these small trees, right? So here's David, who's been instrumental in really identifying these locations. He knew about many of these trees already in these locations. And so we've been going out and pulling samples from these trees to catalog it and working with the American Chestnut Foundation to send genetic material to them, or leaf material to them to do some genotyping. But yeah, these are just small shrubby little things. So far at that particular site at Big Hill Pond State Park in McNairy County, we've identified 6 trees. They're all pretty small, about 1 centimeter to 2.5 centimeters. So, half an inch to an inch in diameter. They co-occur with other species that are typical for what we call mixed hardwood or upland forest here. So these are up on hillsides, typically southern facing, eastern facing, or western facing slopes, very rarely on northern slopes, and up away from sort of the floodplains around rivers and things like that. And at that park, we've mostly found them in the southern portion of that park, south of the Big Hill Pond there. And then, we've visited some other sites, so I'll go walk through a few of these here. So Chickasaw State Park in Chester County, it's similar story, small trees. We've actually found some of them there that were hit by tree fall from other trees, and so they're endangered from these sort of physical events that are associated with wind throw. But we found many more trees there, again, mostly pretty small, but a few of them are getting pretty large and perhaps approaching reproductive age, but it also tends to be the age at which they succumb to blight infection. But again, occurring in these mixed hardwood forests with oaks and beeches and hickories, again, in these upland areas around the lake there at Chickasaw State Park, but also in some other parts of the park. David also identified years ago some trees here right near Somerville in Fayette County, sort of central eastern Fayette County, effectively extending the extant known range of chestnut, right? So these are beyond the historically understood range of the chestnut shown in that gray shading in the map there. So these are really important individuals because one of them is small, but one of them is really quite large. It's getting up to this age at which it could become reproductive, but we're keeping an eye on it also because it could be hit by blight, and that would effectively end its path to being really important for producing flowers and nuts. Other places, the Wolf River Management Area again in Fayette County, southeastern Fayette County. So just south of LaGrange, Tennessee, we found a few trees there. David and some collaborators, Bill Turner and I think Joe Schibig maybe, had also done some searching there. A few of these trees occur on the wildlife management area, again, on these upland slopes. Curiously, we found a couple of stumps there that may be the original trees that were cut or were afflicted by blight and then cut later. It's a persistent wood, it doesn't rot very easily. So they may still be the originals, but hard to identify, covered in moss and things like that. But again, we only visited a few of those that are on the wildlife management area. There's also many more trees that at one point in the early 2000s were documented on some adjacent private land, but I have not been able to go and revisit those. But it does highlight the citizen science component of this too. You may be someone that owns some land or knows someone, maybe your grandparents own a back 40, and it might be worth your while to go out and search that for chestnuts. And then, let us know if you're willing to do that. And this last site is in Holly Springs National Forest in Benton County, Mississippi. And so this is right near the headwaters of the Wolf River, here just upslope actually from Baker's Pond, along part of the trail there, the road that cuts through there. But again, these are getting up there. These are relatively large trees that occur in sort of mixed hardwoods again, although there's a lot of pine plantation around that. And they've been doing some mitigation here, planting in this area associated with recovery from a 2015 tornado that ripped through there, but somehow missed these chestnuts that were just kind of down in a little hollow or a little river drain or creek drainage there, and were spared the fate of being knocked over or destroyed by the tornado. And so you can sort of see that up here. There's sort of a road that goes there and they're down in this little hollow, but the rest of this, and there's a lot of tree fall in there from that tornado ripping through there. And Dr. Craddock had mentioned chinkapin. There's all these other Castanea species around. And what I wanted to just say here is just say that we do have these around in our area, and we have seen some chinkapins around. They're a bit smaller, a little shrubbier in general, but sometimes can be difficult to differentiate from American chestnut. But one of the biggest things for us here in the southwestern part of the range is that they don't really overlap much. So these two Castanea punulas, the chinkapins, are from like Hot Springs in Arkansas and down here in the Tombigbee National Forest in sort of central Mississippi. And it doesn't really get up here too much. So we aren't too worried about mixing these up, at least in our part of the range. And the last thing I want to wrap up with here is that, we want to find these chestnuts. We want to find more of them. And in my lab, we've been focused on trying to predict where we might be able to find more, because we want to go out and collect more of these chestnuts and fill these gaps more, especially in northern Tennessee and other parts of northern Mississippi and other parts of West Tennessee. And so we've been doing some computer modeling using some approaches with ecological niche modeling to build these predictive maps of areas that might be climatically and ecologically suitable for the occurrence of American chestnut based upon some historical records or historical occurrences, which are shown here in with these black dots on the map. And so the red areas and the yellow areas too on this map are showing areas that are predicted to be climatically suitable or otherwise ecologically suitable. So we're including things like temperature and rainfall and topography, but also the biotic components of the environment. So things like whether there is forest there, what is the land use history like in those areas? And it does a pretty good job of predicting the areas. So far, we're still refining this where we do find chestnut, but it also makes some predictions, some areas where we don't currently find chestnut. And that may be for some different reasons associated with the ability to get there or some human usage as well. But so far, these models are actually being strongly influenced by components of the model that include things like canopy cover. There has to be forest there for these to exist in, and also the land cover. So there has to be forest there, not a farm field or a pasture or something like that, as shown in these brown and yellow areas. Those are converted to farm fields or intensive pasturing for livestock. And so you wouldn't tend to find them there. You're going to find them in these little wood lots associated with a farm, or in these forest remnants associated with state parks and county parks and things like that. So I just wanted to wrap up and say, this is an ongoing effort. It involves a lot of citizen science. I've been reaching out to people in the local community, and that's how I found David Smith, who has been really instrumental in making a lot of this work happen. And so, you can help. You can join Wolf River Conservancy. You can join the American Chestnut Foundation, get involved with those programs. You can also download this free app to your phone called iNaturalist. It's put together by academics and people working in nonprofit organizations that makes this really easy to actually go out and explore the environment and to document the presence of species. And some of you may actually be users of iNaturalist, but it's really great because you can say, hey, I found a chestnut on my grandparents' farm or something, take a picture of it, and it logs the location. And that makes it available to other researchers and other community members, and makes it easier to establish those connections. So that's how I found David. That's how I started getting re-involved in American chestnut. So So with that, I'll wrap up. I'm a little over, but I hope that you guys were entertained and learned something today. So thank you to everyone. Thank you to my co-investigators and my collaboration with Hill, the people at the American Chestnut Foundation, my prior funding for some other projects from the National Science Foundation, all the members of my lab that have been working on some of this. And of course, thank you to the Wolf River Conservancy for hosting this and for the invitation. And you can reach me at multiple different modes of communication there. And with that, I'll wrap up and pass it over to, I think, Catherine. So thank you.
Yes, thank you so much to both of you for wonderful, very interesting programs. I do have a few questions. I'll let you guys decide who answers, I guess. Dr. Craddock, you might want to come back, turn your video back on and your microphone so you can answer. The first, I'm going to give you two questions from the same person. First, are water chestnuts in the chestnut groups, or are they something else? And how many trees do you have to cut down to breed 1/15th, 16th American chestnut with blight resistance?
Well, that sounds like a Hill question to me.
So the first one, water chestnuts are not chestnuts. In fact, they're not even trees. The water chestnut, which is popular in East Asian cooking and also in the United States, and is actually a marsh plant. It's the starchy root of a kind of a, I think it's a bulrush. It's a rush that's cultivated in rice paddies and paddy soil. So very different, not related at all. On that same note, neither are buckeyes. Buckeyes, which in many parts of the English-speaking world are called chestnuts, but buckeyes are not chestnuts. They're not good to eat. They're very, very bitter, even toxic. And so, if you went to the UK, if you went to England and said, show me a chestnut tree, they might point to what I would call a buckeye. And if you say, no, the other kind of chestnut tree, they would say, oh, that's a sweet chestnut. So in the UK, what we call a chestnut tree is a sweet chestnut. And, just there are other things. If you go to Australia, they have another thing they call a chestnut. If you grew up in the Caribbean, it might be the seed of a jackfruit. I'm doing like this because the jackfruit is this giant big thing, and the seed of the jackfruit is called châtaigne, which means chestnut in French. And so that's why we botanists learn the botanical names. So Castanea or Castanea is the genus in the Fagaceae, the beech family that includes the chestnut trees that Rob and I are studying. The question about how many trees, years ago, one of my grad students said, Dr. Craddock, does it bother you that almost all the trees you've ever planted are dead?
That would bother me.
Well, it is sad, but on the other hand, I think we're making progress. And I can take you to the field and to the orchard and show you trees that we've bred that look like American chestnut trees, and they have adequate resistance. They can live long enough to bloom and produce a nut. Now, we had some on the campus at Rhodes College that were from the last generation, and I thought they were worth saving. I would have kept those, but apparently the campus landscape guys said, no, they're ugly, and they killed them. So we're going to replace them. I told Rob they came with a lifetime guarantee. But realistically, in the backcross orchards, the selection rate is about 1 out of 8. That's the selection rate. We want to keep diversity at that level. When we get to the F2 generation, which is at the seed orchard level, it's about 1 out of 100. So to get one good one, I'm going to cut down 99. Yeah, and to make any progress at all we have to be pretty ruthless about that. Hopefully, in the coming years, using some, we're waiting for the tricoder, remember from Star Trek, right? You just point it. You think I'm kidding, but they have these things that are like sniffers now. They can detect, your dog, you could train a dog to sniff a resistant chestnut tree. If we could do that, we wouldn't have to kill so many. We could just sniff the nuts and just plant the good ones, and we could eat the rest of them. Next question.
Okay, I think this is a related question. How many generations of trees might it take to get to the ideal resistance? resistant species?
Well, again, I'm sorry, Rob, it's a breeding question.
Yeah, that's all right.
So if you go back to what we call the Burnham plan, Charles Burnham was a member of the National Academy of Sciences, was a corn breeder from the Midwest, who was one of the people who started the American Chestnut Foundation, proposed that we breed chestnut trees like we breed corn. And his plan was a F1, which is the interspecific first hybrid, then a first, second, and a third back cross, and then 2 generations of intercrossing to fix the resistance. And that, if you consider that that's about 10 years between generations, we know now, Burnham's idea was good. It was solid. It was based on good science. The chestnut tree hadn't read the textbook though. And so we know now it's a little more complicated, different from some of the diseases that Burnham was breeding for in corn. The blight resistance in the Chinese chestnut is controlled by many, many genes that are scattered all across the genome. So it's become, I don't want to use the word impossible, but it becomes logistically, operationally, very, very difficult to recover Chinese levels of resistance in the using that plan. So plan B is to complement that with the transgenic approach and or some molecular screening. In other words, we might be able to screen better using molecular markers. In other words, if I had a DNA signal for some or many of these genes, I might be able to, again, I could sample the trees in the nut or in the embryo. We might be able to speed up the breeding that way. But it's a long-term thing. How many generations? I'm optimistic. I think the prognosis is good. We don't need a tree that's 100% immune to chestnut blight. We have also a biocontrol. I mentioned the biocontrol is the hypovirulent strain of the fungus, which is caused by a virus. If you combine hypovirulence and some modicum, even an intermediate level of blight resistance, you can get the trees to live long enough to bloom and produce offspring. And as soon as we've got populations of trees that are blooming and producing offspring, then natural selection can take over and the trees can resume evolving on their own.
I think that's a really important component of that too, is the conservation aspect of it, conserving what we do have and trying to get some of that sampled into those germplasm collections, because we need some of the genetic variants there that may interact differently with those resistance genes that you're breeding in, right? And so that may make the issue easier or harder, but it could make it easier, and it would also help bring in some of those local genes to different parts of the range. And that, even if you get that, as you said, that intermediate level of resistance, that's really important for potentially getting trees that can do the natural cycle. They can get up to being reproductive and then start breeding amongst themselves.
Mm-hmm. Okay, I've got more genetics questions. Is anyone using CRISPR to alter the genetic makeup of chestnut trees to address blight resistance?
I don't know if anybody's using CRISPR. Do you know? Or
well,
is that...
I think yes, we don't see those results yet, but I know that they've got, at the ESF lab at Syracuse, they're working on all of these sort of genetic engineering approaches, as hard as they can. And we've had meetings up there together, and we know they're trying. I haven't seen any results published on CRISPR. Of course, for those who haven't heard of it, CRISPR is a technique where we might be able to edit the genome. It's different from the genetic engineering approach that's been used so far, where we took a gene from a wheat plant and we created a transgenic chestnut tree by inserting a gene from another species into the chestnut tree. In the CRISPR approach, it would be going in there and tweaking the genetic code in a way that would allow the chestnut tree to somehow defend itself. I don't know, here's the terrible admission. We don't know what it is about the Chinese chestnut tree that allows it to survive the chestnut blight. The same fungus, by the way, makes a disease on oak trees too. It's a terrible invasive pathogenic organism. It causes diseases on eucalyptus trees. It's a terrible pathogen. It doesn't affect cabbages. It won't grow on cabbage. You know, we don't know, and there's some basic unanswered biology questions there. And I think CRISPR is a little bit putting the cart before the horse, because I think before you start tinkering with CRISPR, you better know, you know, a little bit more about the basic biology.
Okay. In reference to that wheat gene, how come one can't use the enzyme for southern grown wheat for Tennessee chestnut trees?
To presumably to presumably accentuate the local adaptation to the south, I suppose. I think that oxidase gene that makes that enzyme it's not going to contribute to the local adaptation. It either makes that enzyme or not. Is that right, Hill?
Well, maybe I didn't understand the question, but if that's your interpretation of the question, then Robert, yes, the gene from wheat is just one protein.
Yeah.
It's just the protein. And it's a protein, it's an enzyme, and wheat isn't the only plant that has it. Lots of plants have it. Bananas have it, you know, strawberries have it. Lots of plants make oxalate themselves, and then they destroy their own oxalate. They use oxalate to shuttle, you know, metabolic products from, you know, around. It's sort of a peculiar thing that American chestnut doesn't have this enzyme. And so, when we talk about adaptation to the south, we're talking about things we don't really know anything about. There's some basic assumptions we make, is that these trees have lived here for millions of years before the ice ages, and then they suffered through the ice ages for, you know, hundreds of thousands of years. The assumption is that the genes that they need to do well here in the south are here. And it behooves us to conserve as much of that genetic diversity as possible, because we don't know what will be valuable or what is valuable. But it's certainly not just a protein or another protein. It's a whole suite of genes that interact, and some of them are probably developmental genes. Some of them make real products that work together with other gene products. In other words, it's going to be complicated. And you know, we know from lots of other examples that, you know, I don't know, you don't know what you've got till it's gone. If we lose it, then the question is moot. If we allow them to go extinct or be extirpated from these areas of their natural range, then we've lost it for good. So there's some urgency, I think, in the conservation.
Right.
I don't know if I understood the question, but I tried.
I'm just reading them as they come. Here's another one. Why not experiment with different Eastern breeds outside of Chinese? It probably means Eastern species of chestnuts.
Yeah. Do they all cross with each other? Are they all interfertile?
So all species in the genus have been hybridized artificially. Some of the hybrids are easier because they bloom at the same time. Others of them are more difficult, and apparently there are some other incompatibility problems. For instance, you mentioned that in your part of the world, chinkapin and chestnut don't grow together, but they do in a vast area of the Appalachians. We get
hybridized in those places.
Well, there was some thought that they did, but we actually haven't found any really good evidence of hybridization between the species. They may have in the ancient past. It may be, on the other hand, it may be just some sorting out of ancient diversity. We're just looking at the residue, you know, during the Pleistocene. I really liked your Pleistocene maps, Rob. Those were great. They were pretty much scrunched up together, you know, and now they're spreading out. They have spread out, but they exist. My point is they exist as distinct, discrete species. One is a shrub, one is a tree. And, you know, in oak, related to oak, in oak, we know that there's introgression, natural introgression is frequent in oak. Apparently not so in the chinkapins, though. We've looked and we don't. I've got a paper coming out with Taylor Perkins. You'll see it soon. where we've looked for this signals of hybridization in the chinkapins, and we just don't see it. That doesn't mean it's not there. Doesn't mean it's not there. So other Eastern species, as in East Asian species, we are. So the ones we're interested in are the Japanese chestnut, Castanea crenata, and I showed that work. We've used crenata as a source for resistance to Phytophthora. It's very good. Mollissima, which is the one we call the Chinese chestnut. And then the other species from China that's very interesting to us is Henryi. And Henryi, to begin with, is a timber tree. It's a tall forest tree, different than the Chinese chestnut, which is sort of a spreading shrubby orchard type tree. So the Henryi are very interesting also from that point of view. They vary in blight resistance. And the problem there is that historically, chestnut breeding started in New England and Maryland, and then also in Connecticut. Henryi is not cold hardy. So it won't grow north of Virginia because it just freezes in the winter. But we can grow it in Tennessee and Alabama and Georgia and Mississippi, and you could grow Henryi in Memphis. And that's certainly worth a try. I hope I answered that question. But yeah, we're trying those, not just the one Chinese chestnut. We've got multiple sources of resistance.
That Phytophthora issue, I had seen some references to Phytophthora having been sort of a pest here in the Mid-South at one point in time and may have actually accelerated the decline of chestnut in this region. Do you know anything about that, or?
Well, so the earliest papers on Phytophthora cinnamomi were sort of reconstructing, from anecdotal evidence. It looks like it was here by the 1840s, probably coastal Carolinas and coastal Georgia. It spread throughout the Piedmont. It affects a very, very wide range of plants, unrelated plants, and including chestnut. And by the 1940s, it was basically throughout the agricultural South, up to Kentucky and Virginia. It's been here ever since. It's limited by cold, which means at high elevations in the Carolinas, East Tennessee, and Southwest Virginia, it's not a problem. It was first reported from Pennsylvania about within the last decade, in the Pittsburgh area of Pennsylvania about 5 years ago on chestnut. And so it's spreading. It certainly was here before chestnut blight. And the famous, I say this to my students, don't believe everything you read about chestnut trees, right? Here's my light, I've been sitting still too long. The range map. You guys, Rob, you found chestnut trees beyond the range of American chestnut. Well, that's because Little, the guy who drew that map, his name was Little. Little didn't know. He guessed. He just drew a nice line over there somewhere where it looked like it made a nice line. He was basing his range map was based on herbarium records that he had access to. The herbarium records are incomplete, and he didn't look at all of them, right? And so, yeah, so don't believe everything you hear, but certainly Little's map was drawn after the Phytophthora wave had eliminated chestnut from the coastal plains and the Piedmont.
Yeah.
We have anecdotal evidence that chestnut may have extended all the way to peninsular Florida, coastal Carolinas. It's gone. It was gone by the time folks got around to making herbarium specimens. They weren't doing any, there were people doing botany, but none of the herbarium specimens predate, the middle of the 19th century. So they were gone, they were long gone by then, so.
Okay. I think this is a question for Rob. Can satellite imagery be used to help find these trees? And I'm going to combine that one with, is there any place to see a chestnut in Shelby County that you know of?
Until the fall, you could have come to the Rhodes College campus to see one, but they took that out because it was right in front of the main building there. They didn't want like this dead looking tree there. Sorry, Hill. But But yeah, we'd like to replace that and have an example here in Shelby County. I've been looking hard. I've heard of these reports of chestnuts in Shelby County, and I've looked around a little bit, but I'm just one person and with a few students that are novices at this. So I really need some help. I need people to be out there to look for things that look like American chestnut. I have seen, planted probably Chinese chestnut like Millésima, or something like that, or potentially some hybrid thing in people's yards and in other places around the county. But I have not seen anything that I would bona fide call American chestnut. And that may be that there's been so much development in the area that has kind of wiped out any of the available habitat for it. I mean, there's a few places within Shelby County that seem like they would be good habitat. For example, up at Meeman Shelby Forest on the river, or T.O. Fuller Park on the south end of the city there. There's a few other places, for example, like over at Shelby Farms, where there's some forest. If there might have been some in there, or there may still potentially be some in those areas. But again, it comes down to like, if family members or friends that have a farm in the north part of the county or something, they are the most likely to potentially have some in a little forest plot that they just don't know about right now. And it really requires getting out there, hopefully at a time of the year that's not tick ridden, but getting out there and searching for these things. The satellite imagery can be really useful. I've been looking at some of that to some degree, but really that's useful for identifying these remnant little forest patches that are around. I don't know about using some of these hyperspectral approaches. So people are using things like LiDAR and hyperspectral approaches from satellites and from aerial flyovers to try to identify different species that occur in those forests based on height and the reflectance of different wavelengths of light. So that potentially could be really useful, but it also presupposes that some of these trees are actually exposed to the air, right? So they're canopy species, and most of the chestnuts that we find or that we know about tend to be smaller trees and aren't visible from the air very often.
Rob, let me just add that
Yeah.
a few years ago, I'm on the committee that reviews the grant proposals from the Chestnut Foundation, which, by the way, supports some of this kind of research. And we had a proposal from these guys in Maine who wanted to get a Piper and fly it back and forth with a camera and take pictures and look for chestnut trees. And we didn't fund it.
Okay.
And they of course, they went ahead and did it anyway.
Yeah.
They had some fancy camera attached to a GPS thing, and they flew a grid pattern. So they flew back and forth north and south, and then they flew the same pattern back and forth east and west. And they spent all summer doing this. They not only found American chestnut trees, they found what might be the largest American chestnut tree surviving in North America.
Wow.
And they found it because it was in full bloom.
Wow.
And it
Yeah.
was
You can see the flowers.
85 feet tall. And they could see it. And they had a computer look at all the pictures. And so they used artificial intelligence then to spot the tree in the photos. And then they sent somebody out there to ground truth it. And sure enough, they got out there. It's a tree 20 inches in diameter, 80 feet tall.
Well, we'd be lucky to find one that big, but I mean, I'd be keen to see somebody trying that technique. I just don't know if we have any that big here in Shelby
No,
County.
I don't think so. I mean, I would love to see one.
Yeah.
Yeah, but right.
Yeah. Oh, really cool.
That
So
was
are
in
there
May.
are there any chestnut orchards in West Tennessee?
Are there some in Jackson, near Jackson?
We have planted American chestnut hybrids as far west as Milan.
Okay.
Where's Milan?
Just up the road.
That's a long way from Memphis, but
Yeah.
from where I'm looking, it's practically Memphis.
Yeah,
There's Tennessee, the Tennessee Army National Guard has a training facility there that had some land, and they planted American chestnut trees as part of their Department of Defense Legacy Act. You know, they're supposed to mitigate, you know, they drive tanks around and blow stuff up all the time. And so they're going to mitigate all the damage. So let's plant trees, right? And I've never been there, but I've had reports that the trees lived and some are still alive. So that's the farthest west. We have some small orchards, and gosh, now I'm embarrassed because I'm not looking and I don't know the counties. I think everything we planted though is east of the Tennessee River.
Yeah, that's a bit far. I mean, it's sort of close here, but it's a good hour and a half, 2 hours drive probably. Yeah.
Okay, I'm going to have to make this question. It's a good question, but I'm going to have to make it the last one because we have 20 more in the chat box. But I think we're going to need to stop at 8 o'clock. So
I'll say if there are other questions that you didn't get answered, feel free to reach out to me via email and I can put that up or we can make that available, but I'd love to hear them. But yeah, go ahead.
And you can access the chat box as well if you'd like. So is there any evidence that honeybee colonies in close proximity to chestnut trees will increase resistance to blight? Very interesting.
I didn't understand the question. Kathy, say it one more time.
Is there any evidence that honeybee colonies in close proximity to chestnut trees will increase resistance to blight?
Increase resistance to the blight.
They're moving the pollen around quite a bit. Yeah.
If they move pollen from a disease-resistant tree to a disease-susceptible tree, the offspring, in other words, if you make a fruit with a seed inside, you plant that seed, it could transfer the disease resistance to the offspring. Yes. I should take a giant step back though and say chestnut trees are primarily wind pollinated. And so there's a huge cloud of chestnut pollen that blows up into the sky, and most effective pollination is probably wind. Honeybees will visit the male flowers because they feed on the pollen, but they're not very effective pollinators. They're just not. They're not. The female flower is set up to be a wind pollen. There's no nectary. There's no showy petals. There's no fragrance on the female flower. Now the male flowers are showy, fragrant, and they produce a big reward, which is all that pollen. So we got lots of insects, not just honeybees, but lots of insects visiting chestnut blossoms because they're feeding on the pollen. And they may be pollinating while they walk around. I mean, they're stumbling all over the thing. They're moving pollen for sure. But it's primarily a wind pollinated species. By the way, chestnut honey tastes just like chestnut flowers smell. Now, not everybody likes that smell, but apparently the people that do like it will pay more for that honey. And it's a very dark honey. It looks like molasses. It's has sort of a peculiar odor. If you live near American chestnut or a Chinese chestnut, you know what that smell is.
So are you funding your research by selling that chestnut honey from all the honeybee hives you have set up on the orchards?
So we have a experimental forest in Virginia. It's the Lasagne State Forest, which was planted. It was the largest planting of chestnut hybrids in the 1960s and 70s. Tens of thousands of trees, mostly from the Connecticut breeding program. And they do have commercial beekeepers that keep their honeybees there, and they market that. Yes. I don't know if they're making any money, but they're doing it.
All right. Well, at this point, I'm going to remind everyone that we have recorded this program, so it will be available for future viewing. And please reach out to Dr. Laporte with questions that didn't get answered. And I'm going to turn it back over to Mark. Thank you both very much for a wonderful evening.
Thank you, Jackie.
Thanks a lot, gentlemen. That was really, really great. Wanted to remind all of you to check your chat box. We did put a link in there for sponsoring a tree. So if you can take a look at that and or go to our website and do a tree sponsor, we would love to have y'all's participation in that. Again, thank you to our presenting sponsors and our event sponsors, Brother International Paper. We really appreciate you and all that you've done for the Wolf River Conservancy. So tonight I'll just conclude by saying, from the Wolf River Conservancy, we hope you have a good evening. Thank you for participating, because it's inspirational to have all of you on this Zoom call, this lecture series that really helps us get excited about fulfilling our mission. So thank you so very much, and we'll see y'all at the next one. Thanks.